Good morning. My name is Alexander Diehl. I'm located at the University at Buffalo, and I'm very happy to welcome you all to the CELS 2022 workshop. We have a very nice schedule today, and I'm just going to give a little background to the workshop series. This is the sixth in the series of workshops that devoted to ontologies related to cell biology, cell science. Primarily, these are focused on the cell ontology, but we've had some other talks in the past as well. This year, the organizers are myself and Oliver He of the University of Michigan. And I want to thank the Cell Ontology Working Group in particular, who've been supportive of this effort over the years. And I also want to thank the organizers of the 13th International Conference on Biomedical Ontologies, ICBO 2022, which is happening next week. This workshop is in association with ICBO 2022. The ICBO is located this year at the University of Michigan in Ann Arbor, Michigan, starting on Sunday. Both in-person and virtual participation is still possible. Anyone who hasn't organized themselves to go to Ann Arbor can still sign up for virtual participation, and I encourage you to do so. You have to sign up in order to receive the Zoom links. I would encourage you to go to the ICBO 2022 website to see our program. Sunday and Monday are a set of workshops and tutorials. They're all going to be set up for virtual participation for those of you who can't be there in person. And I think it's going to be a very nice workshop, or a very nice conference. And so I think we should then maybe just get on schedule here. And so I'd like to introduce David Osumi-Sutherland, who's going to be presenting our keynote talk, an overview of the cell ontology and contributing projects. And I'll stop sharing now. David, you can share whenever you're ready.
Thanks, Alex. I have to admit, actually, I've done a slightly different talk to the one advertised. Hope that's okay. But hopefully a slightly more interesting one than just sort of going through the sort of general business of the cell ontology. Anyway, so yeah, the title of my talk is Cell Ontologies, Atlases and Knowledge Graphs. For anyone who doesn't know me, I'm the ontologies lead at EMBL-EBI. Quite heavily involved in the cell ontology as Alex is, and with a whole lot of projects that are sort of connected with it as well. And really looking forward to hearing the rest of the talks in this session. And a big thanks to Alex for inviting me to do this. So I don't usually like these talk outline slides, but it was kind of useful to me in this case to guide it. So basically, there's four sections on this. One is really thinking through and looking at what cell ontologies are good for. Why do we build them? What are they useful for? I think that's sort of informative to questions of how we build them. What do they represent? So a little bit philosophical, cell types versus cell classes, and then some stuff on data-driven cell definitions. And I think this has all kind of become much more important in recent years with the massive development of single cell data. Some stuff on making cell ontologies or the cell ontology easier to use. I think all of our ontologies kind of suffer from difficulties with usability and limited uptake because of it. And then some more on sort of putting it all together in some applications. Okay, so what are cell ontologies good for, if anything? I mean, I think the main, and this is all probably completely familiar ground to everybody at this meeting, I guess the main use case we really have is making data about cells fair, findable, accessible, interoperable, and reusable, and particularly findable, and to some extent reusable. Knowing what your data is about helps you work out how to reintegrate it, make it interoperable and reusable. But in the first instance, just simply finding it. So here's a really simple example of use of the cell ontology on a single cell expression atlas. You can see the single cell atlas has annotated these green cells as Kupfer cells, and they have this really nice anatomogram where the Kupfer cells are colored in and selectable. So these little purple cells, and so the fact that it's annotated means that you could find with searching. You can find by searching with synonyms as well as labels. If you've got your data annotated with this and you want to bridge to something annotated with other ontologies like NCIT or the BTO, which also have terms for these, and we have cross references, etc. And an ID or a universally resolvable ID for the annotation rather than just text. And then obviously the ontology structure itself is also useful in findability because, so for example, the Kupfer cell has located in relationship to the hepatic sinusoid. In Uberon, hepatic sinusoid has these part relationships up to liver. It's also classified as a tissue-resident macrophage. So if I want to find single cell RNA-seq data sets with tissue-resident macrophages or cells in the liver or cells in the hepatic sinusoid, I can use the structure to do that. So far, so pretty well understood, I guess, by pretty much everybody here. I think you can go a bit further than this, and this is very related. Well, so you can start knitting together lots more information to build knowledge graphs and use these ontologies for atlasing. So this is something we do and have done for a long time at Virtual Flybrain. You can see actually a lot of the terms here, a lot of the nodes here are actually terms in the ontology. So the pink ones are neurons in the ontology. There's another type of neuron here. These are brain regions. We have relationships to the brain regions. So this is kind of a simplified view. You're still looking at the OWL version of the ontology, all these existential restrictions just become simple edges. Here's the links to references here with lots of metadata added for the references. And here are a couple of instances. So this is actually individual pieces of data. These are actually neurons depicted in images, and they have some connectomics data associated with them as well. I'll go into a little bit more detail of how we use this kind of thing in the atlasing section at the end, but with some examples. But here's sort of showing the logic of the so here we go. If we were interested in wedge projection neurons, then this is an instance of a wedge projection neuron all the way down. We're interested in things that have some part in the clamp. We know from this ontology structure in this graph that the clamp, the superior clamp here, which this is related to as part of the clamp, so we can find these 55 images that have some part in the clamp and are a type of wedge projection neuron. I think we could probably do a better job of sort of communicating that in the query here, but there's the basic logic, and you can use OWL directly for a lot of this as well. The nice thing about a knowledge graph is then you can start putting things like numbers on edges and using those in queries, which you can't easily do in OWL. You can put lots of other metadata on edges, like links to the actual data. You can so there's a lot of things we can represent in here that we probably wouldn't represent in an ontology, and we can glue lots of different types of data together. For example, we're planning to glue in all of the data that Damien's going to present later on single cell RNA-seq in the fly. Okay, so but there is kind of a pain point here, and I think this is a point of contention. And through this talk, I'm just going to emphasize that there are cases where there are issues that we have that cause conflict and argument about how to build, but they kind of inevitably come out of the different use cases that we have, and there aren't necessarily clean solutions to these. There may be only sort of rough compromises. So one pain point here is this idea of minimal commitment. So when you make an ontology class, it's usually good engineering practice to try to sort of give what's the minimal kind of definition I can give to describe this cell type. cell class maybe, without adding lots of detail. But actually, findability, the more detail you add, especially in the context of an atlas, but the more detail you add, the more you make this findable. So I can find based on this location to the superior clamp, based on any other amount of information that I layer on here. So they sort of tends towards ontology definitions that maybe seem quite long and detailed from the perspective of people trying to build just a reference ontology for annotation. There we go. What do cell ontologies represent? So I thought this was, I kind of been thinking about this quite a bit recently, and I thought it was interesting to sort of give my thoughts on this, and hopefully this is there'll be space for discussion at the end. So I think I would say that cell ontologies are a mix of cell classes and cell types. And no, there isn't necessarily a kind of hard line between these two, but a cell class I'll call, and this fits very nicely with the way we model in OWL. Members of a cell class share some set of intrinsic or extrinsic properties. So you might, for example, define a GABAergic neuron with a soma in layer 1 of the human cortex. So there we go. There's a nice small set of properties that a cell needs to have in order to qualify as being a member of this class. And cells exist with these properties, and we can find them and we can classify them. You don't need to worry about what a cell type is to do that. Some things that you use to classify cells may, have kind of slightly vague relationship to cell typing. Doesn't necessarily matter. You can build nice queryable structures with these classes. Sometimes we argue about whether, it's worth making very abstract sort of classes, combinatorial classes with some relatively abstract set of properties, but it can be useful for querying, and we can classify. So cell type, I think, is something different. So here's what I think a cell type is, or my attempt at defining a cell type anyway. So cells belonging to the same type share a stable core transcriptomic signature that at least partly determines a set of shared structural and functional properties, the intrinsic phenotype. And that cell type often correlates with anatomical location, cellular tissue context, the microenvironment, which may also partly determine the intrinsic phenotypes. And cells belong to the same type, typically they share the same developmental origin and lineage. So there's this very classic figure from various forms of it from Waddington, where he talks about, he imagines development as progressing through this epigenetic landscape. Epigenetics to Waddington meant something different to like methylation and stuff. It just meant something like the transcriptomic state. So there are sort of stable regions of transcriptomic state, and it requires a big push of energy or information to push between different transcriptomic states. So it's not that, cells are all sort of a mush and can have any mixture of properties. I think that's uncontroversial to say. There exist lots of cell types which have a relatively stable core set of properties and transcriptomic signature, etc. Okay, but I think now I'm going to, I probably need more data to back up these assertions, but I think it follows that there are many possible sets of properties like gene expression, morphology, location, function that you could use to uniquely identify a cell type in a particular context. Although actually, when you actually start working with real data, you find that there are almost none that are perfect because of biological noise. Maybe sometimes it's noise that comes from experimental data as well. But if you try and classify a neuron in terms of its synapses, you'll find there's sort of a noisy level of synapses almost everywhere. If you try and classify by morphology, then the odd neuron, which is of that type, has some pathfinding defect and goes a slightly different direction and maybe finds the right target anyway. There's always noise in the system. Whatever you choose, you're never going to be 100% perfect. So a couple of examples. I'm going to talk about this NS forest algorithm later. It finds minimal sets of markers that are necessary and sufficient to define cell types in single-cell RNA-seq data. The most effective combination of the actual result depends a lot on the context. You'll get different markers out if you have slightly different mixes of tissue, and no set of markers is ever 100% accurate. You can get up to some maybe 90%, 98% accuracy with a data set in defining a cell type. You can't, it plateaus out. Yeah, and there are certainly examples in the flyaway, for example, in the optic lobe, where you can identify cell types by their morphology or by their gene expression profile. I'll give some examples of that later. So this brings me on to data-driven cell type classifications. So these are becoming very common. In fact, these are two of these are quite old now. There's huge numbers of these around. So people do some kind of bulk single cell data collection, most famously, I guess, with single cell or single nucleus RNA-seq, or it might be collection of large numbers of cell morphologies, or it might be morphological, it might be functional profiles, and the means for collecting these in ever larger numbers are evolving all the time. And then somebody does some kind of clustering and says, here's a classification that comes from data. So what's the relationship between this and the ontologies we build? So I think the good news is that in some cases, these classifications, at least the leaf nodes that you get out of them, in some cases they fit really well with the sort of classical cell types that we define in our ontologies. So here's the classification of bipolar cells, and actually, this 2016 paper found a couple of new types of retinal bipolar cell based on transcriptomics, but mostly they lined up pretty well. And here's this morphological comparison technique with NBLAST. Most of the leaf nodes, at least, were well-known cell types when applied to regions of the brain that were well analyzed. But things are kind of a little different in mammalian neurobiology, and there the problem is it's just too bloody complex to use the sort of standard approaches. So nobody's ever really come up with a comprehensive classification of cell types in mammalian brains. We've got lots of partial ones, and a major reason for this is it's really hard between two experiments to know whether you're actually looking at the same cell type. It's just too complex to do that. So reconciling information, aligning them to particular cell types is very hard. There might be functional recordings, morphology, etc. So single cell transcriptomics comes along, and all of a sudden we can take a whole brain region and soon whole brains. In fact, there's papers in development right now on the whole mouse brain, and you can generate a comprehensive data-driven classification of all the cells. So great, we've now got a solution to this problem. For years we haven't been able to define all the cell types in the brain, and here it looks like there's a solution. And even better, previously the problem was how do we know when we're looking at the same cell type? Well, here if you use this single cell or single nucleus RNA-seq data consensus clustering as a I can see. Are there questions coming up? Okay, so maybe feel free to interrupt. I think I'm not doing too bad for time. Where was I? Yeah, so this single cell RNA-seq analysis clustering, hierarchical clustering, can actually be an anchor that we can use to attach all sorts of other types of data when they come from these techniques, which are multimodal transcriptomic techniques. So in PatchSeq, for example, sorry, this is a really bad image. You can get both morphology, function, electrophysiological function, and single cell transcriptomics from a single cell. EpiRetroSeq, you can get a tracing of the projections. as well as epigenetic profiling. Merfish, you can look at 500 genes at once. So, you could develop a decent set of probes based on the cell types here, do Merfish, and then you can see where the different cell types are. So this is great. We can actually start to pull all of this type of information together using single cell as an anchor. So I think in this case, this is the way the whole field's gone. We've got no choice but to try and reflect this. Otherwise, the cell ontology, except for very, very general classification, starts to become irrelevant, I think. If you want to have a bunch of terms that are coming from this kind of approach that people can use to annotate their data for fair data sharing, etc., then we need to be involved. What's a data-driven cell type definition? So a definition of a cell type by direct reference to some specific data. So that could be transcriptomic data for cells clustered by transcriptomic similarity, could be morphological, could be functional, could potentially even be some mix of those. Data-driven cell type classification. So there's actually a classification hierarchy that comes out of the data-driven cell typing. I think this is kind of a bit of a mixed blessing, but we kind of have to work with it. Working with Sean and Hussein in particular, we worked on making this brain data standards ontology. We take this hierarchical clustering of single cell data, convert this into OWL individuals and relate them by subcluster of, and we use this design pattern with a property chain basically to be able to infer subcluster of links. So to be able to infer classifications between classes defined with reference to these cell types. So C1 is defined as having this cluster rather, or cell set. This is C3 is defined as having this exemplar. The design pattern basically allows us to take those hierarchical subcluster of relationships and use those to infer subclassing. So we can basically transfer this hierarchy into a class hierarchy, something much more familiar and usable in OWL. And then over the top of that, we can start layering on different classifications and properties to these nodes, often based on these cross-modal approaces. So we've got some examples here. You can see ET here refers to extratelencephalic projecting. That partly comes from, and you can see some more specific ones, my projecting here, for example. This partly comes from the...
Oh, I just had a question. I think this is really interesting. So are you treating the clusters as individuals, and then?
Yeah, we're treating the clusters as individuals. Yeah.
Okay.
And then we could go further down to the individual cells and have some kind of member of relationship. But basically that's on the data side right now. You have a big cell by gene matrix, and each of the cells in the cell by gene matrix has a cluster identifier, and we're working on. We already have unique identifiers for those, and we're working on making IRIs for them.
Yeah, and are you, at least in the OWL, do you have data properties to hold some of this data too, or would that?
No, we actually have avoided using data properties so far. Well, we haven't really had the need to use data properties so far. So far, this is all some quantification, and I think there's interesting possibilities for using data properties in a lot of this, but yeah, we haven't really explored it yet, and we haven't had a need with what we've been building so far.
All right, thank you.
Sure. Okay. Yep. So we can layer on classification and properties based on this mapping. So, if you have epiretroseq data, sorry, this awful image here, which shows a projection pattern. Actually, you can see some nice projection patterns here. We're really keen, and we're not doing so yet. We're really keen to be able to annotate these projection patterns directly. And we end up with quite a usable ontology as a result of this. All of this is part of the provisional cell ontology right now. And I think it needs to remain there, at least for now. You know, the reference data sets may change. The understanding of cell types may change as more types of data come in. I think especially as developmental data starts to come in. And there are some dangers here as well. So,
David,
yeah.
can you just quickly tell us about the provisional cell ontology in case some people haven't heard of that?
In the provisional cell ontology?
Yeah, what it is.
Yeah, so well, so it's an extension of the cell ontology, and it's a place to put cell types we're just not as certain about right now. I think there isn't a very clean line dividing the cell ontology from the provisional cell ontology. I think that's kind of inevitable. I think that some of the stuff in the provisional cell ontology will come in based on because it's based on limited data. Right now, there are no data-driven cell type definitions in CL. Everything has sort of a classical or some definition in the cell ontology right now. So all of the things with these data-driven cell typings for mammals are going into PCL right now.
David, so I mean, yeah, I also got a question after remote. Obviously, data-driven approach, if you map this to ontologies, it requires that the classes are being generated by different labs are reproducible, right? Have you reached a situation where you can say, okay, if you have a minimal set of positive and negative markers, let's say, if you see this thing in a cluster, then it is a cell type? Or are we already in such situations that we can do that?
So actually, you can sort of work back to give things that are much closer to necessary and sufficient definitions. And maybe I should have emphasized this a bit more. Actually, we have this. I have a slide later on this NS forest technique where you can actually pick out a minimal set of markers that give you, 95 or 98% chance of identifying cells in the cluster. That's relative to the data set, but then if your data set is the whole brain, potentially you can come up with a, necessary and sufficient up to 95% accuracy, 98% accuracy for a set of markers for identifying the cell type. So that gives you a much better mapping onto sort of classical AL definitions.
I see. And how comparable are if you analyze single cell data, let's say from the whole brain from different labs? Same stage.
I don't think we have that. We don't have that yet. We do have data for one brain region from a bunch of different labs, and it's pretty closely comparable, and then they put it all together and made a consensus, which is actually what we built from. There's like 12 data sets underlying this consensus.
Right. Yeah. Okay.
9 single cell sets. Okay. Right. Yeah. But I think the data-driven classification is dangerous, though. Especially, I think, maybe especially because it's based just on transcriptomic data, which might not give us all of the kind of signals for properties. There might not be a signature of the properties that we're interested in that data in every case. And usually these clusterings are simple single inheritance clusterings as well. So here's one example where we had a mapping that really looks like 2 cell types. One, they're both p-val GABAergic cortical interneurons. They're in 2 different layers, and they have 2 different morphologies depending on the layer that they're in. That to me is a good indication that, functionally these are probably going to be pretty different when it actually comes to circuit function, for example. But they both sort of get glommed onto one grouping class, and we don't know which of the subclasses they apply to. We don't have the resolution in the data. It may be that, that cell types are much fuzzier than we think. But my interpretation of this is we don't have the resolution in a lot of the transcriptomics data right now to finally divide these things up accurately. And that brings me on to this. So functionally important cell properties might not be reflected in transcriptomics data, and I think we have some pretty good indications of this. Here's 2 examples. One coming from a paper by Erzel last year on the optic lobe transcriptome in flies, and you can see earlier on you actually get finer cell typing. Actually, we don't have, I think I'm missing a bit here. We don't have it all the way to the adult. by the time you get to the adult, you don't have as much resolution as you have at earlier stages of cell typing. And I think this is probably where, and classically, these cell types are defined by shape and connectivity, morphology and connectivity. And it seems pretty obvious why. There are actual things involved in axon pathfinding, layer-specific targeting, synaptogenesis that are expressed earlier, and they're not needed in the adult. They're gone in the adult. So actually, the things which set up the shape and connectivity of the cells, which are really important for function, there's little or no trace in the transcriptome. Actually, they can get around this with clever systems using machine learning, which then project up to the adult and find sort of minimal signatures and stuff. So they can get around it by mixing in the developmental data. But the adult data alone is not sufficient to resolve these different cell types. And, sometimes the morphology and functional characteristics are shaped by the microenvironment, and that's not necessarily through any mechanism acting through the transcriptome, through changes in gene expression. So very famously, classically, glial cell wrapping of neurons affects the diameter of the axon, which affects the electrophysiological properties and affects the clustering of ion channels along the axon. That's actually turns out to be true in flies as well as in invertebrates. So, things that could be really important for typing are not necessarily always going to be visible in the transcriptome. Big advantage of data-driven, or at least data-linked cell typing, I should say. In VFB, we have links to data where we sort of have de facto standard reference data rather than this sort of formal definition with reference to data. But there are large and increasing numbers of BLAST-like algorithms that you can then use to try and identify the same cell type in different data or similar cell types. Here's one developed by Marta Costa, who used to be part of VFB, and colleagues, where you're actually you skeletonize a neuron, split it into vectors, compare the location and direction of each vector, and some will use all of those pairwise scores, to develop a pairwise score of similarity between two neurons. Works pretty well in lots of cases. Here's some examples with single cell data, SCMap, NS-Forest. There's another one called Azimuth that's widely used. So I think this is really, and this actually, you build this into atlases and you've got ways to start getting the annotation with cell types out there automatically without having the bottleneck of humans looking or manually annotating. Okay, bottlenecks. How can we make using cell ontologies easier? Few things that my group's been working on in various projects. One that we're using very widely now is this concept of semantic tags. A semantic tag is really just a text tag attached to a class or an individual. So we haven't been, a lot of this potentially you could do with adding defined classes and stuff, but what this just gives you a very, very simple tag that consistently, and it allows you to sort of use the power of the semantics, but hack it a little bit as well. So, classically, the nervous system doesn't contain the sense organs are not part of the nervous system, but for grouping purposes, for driving virtual flybrain, we wanted to group them together. So we can use the power of OWL to say, okay, I'm going to put the dopaminergic tag on all these neurons. I'm going to put nervous system tag on here. I want to be able to tag everything that overlaps, has some part in the larva. So I'll put a larva tag on that. There's a danger here, you can deliberately overload these. They could be ambiguous, but we found this an extremely powerful way of working very rapidly to actually develop applications where you use the power of the semantics but simplify it as well. Here's the virtual flybrain knowledge graph, and now you can see semantic tags on here. So this is synaptic neuropil domain, that's a brain region. This is a serotonergic neuron. You can see whether the neurons have any connectivity data on it. Well, it's actually a Sparkle query. So we drive a lot of the application using these Here you can see in Virtual Flybrain some of the tags. The tags get used, show up in search, so you can tell that these are adult cells. We can probably clean up the redundancy in here a little bit more, but you can see on this neuron that it's cholinergic, that it's in the olfactory system. We tag these in this circuit browser we've got, and you can use them for filters when you search as well. Here's another example, mammalian primary motor cortex mini atlas knowledge graph. So this is related to the work I was showing earlier on the brain data standards. This is sort of the knowledge graph version, and that's being used in this case for faceted browsing. So you can click on this semantic tag chandelier cell, and you can use that to find all the other chandelier cells in the knowledge graph. So yeah, again, more work from Hussein and Sean. How can we make using cell ontologies easier? Well, another thing that we're starting to do quite a bit of is generating ontology views. So actually this doesn't use cell ontology here. This is an example from a bit of Uberon, but say a user wants only a limited set of terms, but wants to know all of what are they, and have an accurate representation of all the relationships between them. And here's a reduced view generated by specifying a small number of edge types, so actually subclass of and part of, and a smaller number of terms that were needed. So these are the ones in red. Anita's worked on this extensively for HubMap and the EBI single cell atlas, and Hussein has implemented it in Robot. And I've just realized, I should apologize, I've missed a slide out here which has the details of this, actually how this works. So this is actually using Ubergraph, which some of you may be aware of from Jim Balhoff, that actually makes takes all of the existential restrictions in an ontology and then makes a complete set of infers or materialize all those inferred existential restrictions as a simple graph, as simple triples. So actually what we can do is use that as a source of these to find what are the direct links to reduce the graph. And the relation graph software that Jim built for Ubergraph is also what underlies how this works in the Robot implementation, which is not out yet, but should be out soon. So here's an example of that in action, where the Human Reference Atlas project for HubMap, they only want adult terms. They don't want lots of developmental terms. There's a lot of developmental terms in here, and they only want terms that apply to humans as well. And there are terms in here that apply to other species and not to humans. So given a smaller subset, we can take this really complex graph and we can reduce it down to some visualization that's much more manageable and still gives accurate groupings of terms. Okay. Another one, really, really simple, but I think really important, is the use of symbols. So I think probably most of you who work with ontologies or cell ontologies in particular are very aware of this. So ontologies just require these really long names to disambiguate, because your cell type might be defined in the context of an ontology that has muscles and cells from the tracheal system if you're in flies, and cells from the nervous system. And then if you go with the sort of short names you find in papers, they're way too ambiguous. But users hate these long names. They find them so confusing. They find them really quite intimidating, and they like to just use nice little symbols that can be disambiguated by context, and they will always continue to use those. And we tend to add those symbols as synonyms, although sometimes we don't even add those, because maybe the same short symbol is used for two different cell types, and we think that's a bad thing for text mining. But then we've gone even further away from our regular biologist users. So I think the solution here is adding symbols for every cell type, where you can come up with some standard or some way to add a symbol. I think it's worthwhile doing it. So we've tried to do this extensively for FlyBase, difficult questions. How broadly should we try to enforce uniqueness on these? You know, I think in VFB, we try to enforce uniqueness, at least within the nervous system. Okay. I'm probably running way over time. I'm just going to very, very briefly mention this cell annotation platform that my group's involved with, that it labels fair sharing of single-cell RNA-seq data, and it allows users to, the community to annotate data
David,
themselves.
you do have 12 minutes left, including time for questions.
Okay. Okay, yeah, I'm going to skip through this quick. So, yeah, this actually, what we've tried to do with this, with all of the different fields here, is we've used semantic tags to come up with ways to restrict search. And one of the interesting things is the addition of contextual search. So we can use semantic tagging to, for example, to tailor these autocompletes for both species and region. And so if you type epithelial in the context of the organ being kidney, you'll get, as well as the generic term, you'll get higher hits for epithelial cells that are in the kidney. This is somewhat experimental, but at least the species tagging actually is even more important. So in this case, this is a multi-species annotation system. We can use, we actually have fly data in here. We don't want the fly terms to turn up when people are searching in mammals, and we don't want the vice versa. So we can actually set a filter based on those species terms. In the case of, and we're working on bringing in using the species, the taxon restrictions in CL and Uberon to actually add those restrictions in, or boost at least things, appropriate terms from the right species for other species. Okay, so, and that's quickly. So that's Uga in my group is doing this work along with Bradley, who doesn't like his photo shared. And okay, so it's actually not too bad. So I was just gonna quickly go through, and I think then we can have a little discussion of bringing this all together in virtual FlyBrain. So in this case, we've got lots of we can actually view data on single cells here that we have, and here's the ontology annotation of them. Here's the semantic tags, various bits of metadata and extra information, queries that are tailored to the term itself, a browser for this which is built automatically for any brain that we load up with annotations. It just automatically builds a tree for us using the knowledge graph. And yep, so we can do queries like find all the wedge projection neurons. I mean, that's not too different from any other sort of basic fair usage. We can also, because we have connectomics data in here, we can actually do connectomics queries. So this is finding the shortest paths between two specific neurons. And you can see the tags up here as well, the semantic tags. I already went over MBLAST, so I can skip this slide, but MBLAST is incorporated. We have about 100 and I think we have a matrix of around 160,000 now. Pairwise blast scores just trimmed down to be sparse so that we only have the high scores in the graph. And then you can use that, for example. So here's an untyped neuron from a data set. You can run a blast with it and get a high confidence score to this particular neuron. Actually, this was run the other way around, but we could have run it with the unknown neuron. And this types it as a WPNB1, which is the nice little symbol, or the super long ontology name tier 1 bilaterally projecting wing sensitive wedge projection neuron. And you can see the overlay of the two here. So I think, you know, this is going to be increasingly important and powerful as we get more and more data sets loaded in where we have no cell typing or very little cell typing. So we'll have very large amounts of connectomics data that we're about to load from one particular project, which has very limited typing. So I think being able to start trying to predict types using this is really useful. And there's probably a bunch of really cool things you can do with a knowledge graph once you've got all these numbers on the edges. So some quick acknowledgements. Virtual FlyBrain project. Claire Pilgrim, I think, is here today. You can see on the right in this picture is the ontologist for Virtual FlyBrain. So she's done all the hard work of pulling all that together. And Alex is another curator, and Robbie is the senior developer on that. Yeah, Hussein also works on this project. This was taken at a nice meeting in France we had last week. Okay, I think actually I already covered this. The reason I put this at the end was I think, so right now we're beginning a project where we're starting to pull together lots more of this brain data. And I think this is a great opportunity to start building knowledge graphs that start actually pulling in the data itself. So in this case, there was a lot of work that went into kind of pulling all the data together for this cell type knowledge explorer pretty much manually. But, you know, there's no reason that lots of the data sets, as they're being annotated, shouldn't also get persistent URLs, which can be used to resolve them. And then we can have much more effective linked open data system, which allows you to find the data using ontology terms and using the structure of the graph, but then actually pull the data back directly and combine it into your notebook sessions and do your analysis, etc. So that's what I hope is the future. Okay, and lots of people are involved in that project. Okay, so maybe I'll take some questions. Thank you.
Thank you very much, David. I think this was a really, really nice overview of many of the issues in how we can build accurate cell ontologies. One question I have is that a lot of what you present is sort of on the edge of cell type or cell information in the form of a database versus what we can really put into an ontology. And I can almost envision a system where you'd have a database and depending on what differential you're interested in, you could sort of build an ad hoc ontology on the fly, almost in an analogous way to the way you showed those graphs where you like Uberon, where you took out some intermediate terms because they weren't needed for the representation you were interested in.
Yeah, I suppose it's possible. I'm not quite sure. Sure. I mean, if you wanted to directly use an ontology representation in that way, I didn't really go into the details of the pipeline that we've got to convert between knowledge graphs, and I mean, there's lots of them around as well. Chris Mungle has one he uses a lot. Yeah, lots of different groups have developed them. This particular one is really developed to make a very easily queryable and readable knowledge graph. So all the edges have human readable names and stuff, but it's lossy as well. It only supports a limited subset of OWL, so it doesn't bother to try to encode equivalent class or nested class axioms. It's really just interested in existentials. Yeah, it can do anything that's like just a binary pair of, you know, a relation and two OWL entities. So it can do value restrictions as well, for example. But
this is Martin again. Can I ask a question?
Mm-hmm.
So actually, I don't know if this is a question more for Alex or more for David, but as you might know, I'm responsible for the gene expression database in the mouse, right? And we annotated a lot of terms to the anatomical ontology that we developed together with our colleagues in EMBL. And we are now starting to annotate cell types too by combining anatomy terms and cell type terms. And so what we are doing, and I think what many people are doing, will start to annotate cell types, is that they rely on the cell ontologies. That is an Arbor, right? So my understanding is that what you are doing here is a very new approach, actually, to develop a new, a new representation of cell type information. Is that correct? I mean, what does this have to do with? I guess my question is, we are in a very different, I feel we are in a very different place. You know, and most people who are starting to annotate cell types for community resources rely on what is in Arbor. So
yeah, no. So actually, we added lots of terms to the cell ontology we're not releasing a Arbor version of the provisional cell ontology, but the cell ontology itself is released in Arbor. And we added lots of terms, and we are adding lots of terms that are sort of more general terms for cell types to the cell ontology as part of this work. So Sean Tan, who's here, has added many cell types to the cell ontology. Those are all visible in the Arbor version. So for legacy reasons, we keep an Arbor version release, really, but the cell ontology itself has been developed in OWL for quite a few years now. So really, we're not really that radically different from anything we've done before, and we still try to support the legacy uses. I would say I wonder how much manual annotation of markers for cell types is going to start being swamped by all of the single cell data, but other people on this call could probably talk more sensibly about that.
Yeah, I'm just wondering what we should, what's the best approach for us would be, to annotate cell types. That's where I'm coming from, right? And there's also another thing that I really liked in your talk, which was that you described cell types and say they are located in certain anatomical structures, right? Which basically you have the anatomy here and you have the cell type ontology there, and you combine those, which I think was the original idea, to combine anatomy and cell types to describe, the thing. If I currently look at the legacy cell ontology, right, it seems that the anatomy is folded into the cell ontology. So that means you basically have to put in all the anatomy from all species and add cell types to them, which doesn't make sense to me. So I think this tagging, I really prefer the semantic tagging approach that you are using, because it's just, I think it simplifies things a lot.
Yeah, I mean, the idea is to give a sort of simplified way of gross classification to an approximate, careful about the word classification there, but for application purposes. I think that, the Uberon and the cell ontology have been linked together for many years already, and we're just expanding the number of links. And if you have lots of annotation with the mouse anatomy ontology, then we can bridge that, because Uberon has all the cross references. So I think it's possible to use the infrastructure that we've got to continue extending what you do.
Maybe we can talk at another time, a bit more in detail about those things. Thank you.
You're welcome.
Okay, I think we're going to need to move on to stay on schedule, but we do have a period after all the talks where we can have additional discussion and questions for any of the speakers. So thank you very much, David. So our next talk is called Summarizing Single-Cell RNA-seq Expression Data in FlyBase by Damien Goutte-Gattat. I'm sorry if I've ruined your name, but please, Damien, go ahead and share your screen.
Damien,
sorry about that. Trying again.
Oh, there you go.
Sorry. Good morning or good afternoon to you all. So I'm Damien Goutte-Gattat. I'm a single-cell RNA-seq curator at FlyBase. And today I will present you the work that we do at FlyBase to exploit single-cell RNA-seq expression data in our database. I will explain a little bit also, even though this is not really the main focus of this talk, how this relates to the ontological work that we do in FlyBase with our Drosophila anatomy ontology. So very quickly, because I assume most of you are probably already familiar with that. So FlyBase is the model organism database for all data related to Drosophila, especially Drosophila melanogaster. So it has now reached its 30th years, even though if we take into account the non-digital version, we can trace its history back to 1934 or so. And it is maintained by 4 groups spread between Cambridge in the UK and 3 different universities in the US. So about single-cell RNA sequencing, this is a technique, or more broadly, a set of techniques that have been introduced in mouse slightly more than 10 years ago. It has been used in flies since 2017, and its use is increasing as we speak. We currently have approximately nearly 100 single-cell RNA-seq papers in flies as of last month, including one major paper which has been published this spring, which was a fly cell atlas, so a complete study of the entire mapping at the single-cell level of the entire adult fly. And we are currently witnessing at FlyBase a pace of about 3 to 6 new single-cell RNA-seq papers every month. So all of this generates a huge amount of data. And one problem with that is that those data are not necessarily easy to exploit, as was exemplified a few weeks ago by this tweet, by this real tweet that I took from Twitter of fly researchers asking, basically saying that yeah, all those single-cell RNA-seq experiments are quite nice, they're quite cool, but if we are not ourselves RNA-seq people, what can we do with those data beyond just looking at some fancy UMAP or t-SNE plots? And so one role of the model organism database, such as FlyBase, is to help users to exploit the kind of data generated by single-cell RNA-seq experiments. So what are our aims at FlyBase regarding single-cell RNA-seq data? We have mostly 3 distinct aims. One is to help our users discover which fly single-cell RNA-seq datasets are available, get some information, some metadata about those datasets, and this will be the focus on this of this talk. We also provide our users, our FlyBase users, with a quick overview of the most important expression data from those datasets. Which mostly means that we want our users to be able to find directly on our websites without having to go look at the full complete set of data. We want them to be able to find some answers to some questions, such as what are the cell types in which a given gene is expressed based on what has been found in single-cell RNA-seq data, and in cells in which my gene of interest is expressed, what is the proportion of cells in which this gene is expressed? Is it expressed in 100% of cells of that type, or is it expressed only in 10%, 30% or so? And in those cells in which it is actually expressed, what is the average level of expression? Is it lowly expressed, or is it expressed at a very high level? So this is the kind of answers we want our users to be able to find answers for directly on flybase.org. So in order to do that, we have set up a collaboration with the EMBL-EBI Single-Cell Expression Atlas, which is both a database and a website that collects and analyzes all single-cell, or most single-cell RNA-seq datasets, both in flies and also in other species. So we are relying on the EBI SCEA curators to validate the datasets that we found in the literature, to process those So very briefly, I was talking about the collaboration that we have with the EBI. So basically, the EBI is providing us with, they validate the data sets that we found for them. They process them using a standardized pipeline, store them for us so that we can direct our users to those data sets when they want to get the full data. And they also provide some visual tools, so they allow users to create their own UMAP or t-SNE plots to explore the data sets as they want. And what we do on FlyBase, so we discover the data set in the first place, even though I will not talk about that today. We validate the cell type annotations that we get from the single-cell RNA-seq authors before the dataset is loaded on the SCEA. And we summarize the expression data so that we can then use that the summarized data to provide our users with answers to the questions I highlighted before. So a quick overview of this pipeline that we have between FlyBase and the EBI. So everything starts when authors of single-cell RNA-seq papers upload their data to a data store such as the NCBI Gene Expression Omnibus or the EBI Array Express. From there, their data is picked up by the EBI data curators where they process the data using their standardized pipeline. And in parallel, FlyBase curators, that is mostly myself, we get the cell type annotations from the upstream authors. We validate those cell type annotations against the Drosophila anatomy ontology, and I will talk a little bit more about that later. And we provide the validated and converted annotations to the EBI curators so that they can then integrate them with the data that they got from the data stores. So then all of that, the cell type annotations and the raw data is processed by the EBI. And at the end of their pipeline, they published an analyzed version of the dataset that we can then pick up directly from the SCE website. We can then summarize this dataset, and I will describe that in the next slide. And this summarized version of the dataset, this is what we store in our database, and this is what we use to power our website. And the summarized version of the data that we store is also picked up by virtual FlyBrain curators, and this is basically picked up from our Shadow database and loaded into the graph database that is at the core of virtual FlyBrain, and that is used then to power some custom displays on the VFB website. So what do I mean by expression data summarization? So the kind of data that we get after single-cell RNA-seq, after a single-cell RNA-seq experiment is an expression table, which is basically a huge table where each column represent a single cell and each row represents a single gene. And so the EBI is providing us with two versions of those tables. One which is the raw counts table, so in which each tile in this table contain the number of reads aligned to a given gene in a given cell, and the normalized counts table, which is basically the same except that the value is expressed in counts per millions of mapped threads. So this is the raw data that we are working with, and the summarization of this table is basically for each couple of a gene and a cell type represented as an ontological term from the Drosophila anatomy ontology. We extract from the raw table two numerical values. One is the extent of expression, so that's the proportion of cells of that cell type in which the gene has been detected at all, at whatever level. This is computed as the number of cells in a given cluster with a non-zero read count divided by the total number of cells in the cluster. And the second value that we extract is the average expression. So the average count per mapped threads in cells of that type that do express the gene that we are looking at. And so in the end, we store in our FlyBase database a table where rows would look like this. So for each row, we have a gene identifier. So for example, here, FBGN 17. A dataset ID which will allow us to make a link with the metadata that we store for a given single-cell RNA-seq dataset. The cell type of the cell type of the cluster, cell type that has been identified for a given cluster, and the two values, the extent of expression and the average expression of this gene FBGN17 into in cells of the given type. So those two values, extent of expression and average of expression, are extracted from the expression tables that are provided to us by the SCEA. So now the question is, what about the cell types? Where do we get this cell type information? We get that from the cell type annotations that are provided to us directly by the author of a single cell RNA-seq papers. And what we need to do before we can actually fill this case here, this cell in this table, is to validate the cell type annotations that we get from the authors, to convert them into ontological terms representing cell types from the Drosophila anatomy ontology. So the reason why we need to do this validation conversion is because typically the annotations that are provided to us by the authors will not directly correspond to cell type terms in the Drosophila anatomy ontology. So there are several reasons why this might be the case. The most common one is simply because author will use what they think is a common name for a cell type, but not necessarily knowing that in the ontology, this cell type is actually known under another name. So an example of that, for example, is astrocyte in brain samples. People will routinely speak of astrocytes when they want to speak about non-neuronal cells in the brain. But in the Drosophila anatomy ontology, we do not actually have a term for astrocytes. The correct name, the official name for those cells is actually astrocyte-like glial cells, because we consider that astrocyte is actually a vertebrate term, so we do not call them astrocytes per se in the ontology. So this is by far the most common type of conversion that needs to be done. Another quite common occurrence is a case where author may use annotations that refer either to cell states or to a mix of cell types and cell states. So one example, for example, here was an annotation where people have annotated a cluster as being a cluster made of plasmatocytes prolif, where we have both a cell type, so plasmatocyte, which is a type of blood cells in Drosophila, and prolif, meaning that they are proliferating plasmatocytes. So we cannot use that directly because we do not represent, at least for now, we do not represent cell states in the Drosophila anatomy ontology. It's an anatomy ontology, so we represent cell types, but not cell states. So in this particular case, we need to convert that to plasmatocytes only, and we lose, at least for the time being, we lose the part of the information regarding the cell state. Another case is for clusters that has been annotated in such a way that cluster in which people, the author, were not able to identify one precise cell type. So instead, they identify the cluster as being a mix of different cell types. So for example, here, a cluster that had been annotated. Just to present all those examples, in case it's not clear, those are real-world examples from data sets that we have curated in FlyBase. So we had a cluster that had been annotated as olfactory projection neuron innervating DA1, VA1D, or DC3 glomerulus. So in the Drosophila anatomy ontology, those are three distinct terms. We have one term for OPN innervating DA1, one term for OPN innervating VN1D, and one other term for OPN innervating the DC3 glomerulus. Another yet kind of example is when clusters are annotated using a term that represents an organ or a tissue rather than the cells that make up that organ or this tissue. So for example, a cluster that has been annotated as dorsal vessel instead of cardiac cell. Another example, we have actually briefly touched that in the discussion after David's talk, is for the case of a cluster made of cells that are cells of a generic type, so for example, neuron or epithelial cell, that happens to be located in one particular organ or one particular tissue. So here, most often what needs to be done is not actually to correct the notation itself is correct, but we may not at the beginning have the corresponding term in the ontology. So in this case, what needs to be done on the FlyBase side is to actually add the missing term for this particular type of cell in this particular location or in this particular tissue. And finally, the last two cases are the case of clusters in which the author were not able to precisely identify a cell type. So what I called an uncertain identification. So for example, in this example, BTL GAL4 positive likely to be ovary cell. That's not really a positive identification. And also a case coming from a data set in which the author had studied blood cells. There was a cluster in which the only thing that author were able to say was that this cluster is made of cells that are not hemocytes. They can be anything else, but they are not hemocytes. So the conversion that we do in this case, actually, we do not do any conversion at all. We basically exclude those clusters from the data summarization that we do, because if we do not have a precise cell type that we can attach to a cluster, then we cannot show any kind of expression data for that cluster. So the cluster will still exist in the dataset as stored at the EBI, but it will not show up on FlyBase when we show the summarized version of the data. One last thing that I want to explain here, and quite importantly, is that when I say conversion, this is actually only adding a new version of the notations that are using terms from the Drosophila anatomy ontology. We do not, we keep the original annotations in place. We do not replace them by ontological terms. So when you get the data set on the EBI website, you will find both the original annotations from the author, so you will find that under the name author labels, and you will also find the ontological version of those annotations. So after application of all the conversion that I described above, and you will find that under the name ontology labels. So we do. So basically, this conversion process is non-destructive. We keep the original annotations at all times. So no, very briefly, because I already took more time than wanted, but arguably this was not entirely my fault. I'm sorry. So what is the visible output on the FlyBase website? One visible output is the dataset reports, which are actually not very interesting for us. This is just where we put all the metadata that we have captured about the dataset, so that people can know about some general information about the dataset, such as which strain has been used to obtain the data, which methods has been used, etc. And the most important visual output that we have for now is a recent addition to our website, which we call the cell types ribbon, which directly use the summarized data that I've described in this talk. So that's a new type of ribbon that we've added to our gene report page. So this is displayed on the report page for every single gene. And this shows a selection of 20 major cell types from the Drosophila anatomy ontology. And for each cell types, the corresponding tile is colored based on the extent of expression for the gene for the current gene in that corresponding cell type, based on the data from the Fly Cell Atlas dataset that I mentioned earlier. So this big study of the entire adult fly. So this is the main visible output that we have for now. So some things that will give our users straight from the gene report page an overview of which cell types is a given gene have a given gene been found to be expressed. We are envisioning more complex displays to add to our report page for last year, but this is still work in progress, and we are still not committed to making those kind of displays. We are currently waiting to get user feedback from our users so that they can help us decide which kind of displays will be the most useful for them. And very briefly again, I mentioned at the beginning that our summarized data is also used by virtual FlyBrain. So this is what it could look like in future releases of the virtual FlyBrain. So you have here on the left a list of all, if you are on the page corresponding to a given neuronal cell type, you can find all the clusters that we have in FlyBase in which the corresponding neuron has been identified. And from there, you can get a list of all the genes that has been found to be expressed in clusters, in a given clusters of a given cell type. And that's it. So apologies again for the hiccup during this presentation. So hopefully this was still useful for you. And of course, I welcome your questions if we still have time for some questions.
Thank you very much, Damien. It's unfortunate, but I think we can still give you maybe 1 or 2 questions. Would anyone like to ask Damien a question? I think these are very nice tools you're using in your interface to display the data, and I think that adoption of the ribbon is a very nice tool. I mean, I know that started, I think, with the gene ontology, or maybe the model databases, and I think it's very important that we have these sorts of visualizations to make the data accessible to people who maybe can't drill down into the full data.
Yes, that's exactly our aim here. Basically, the idea is that we provide directly on FlyBase quick information for users who will be satisfied with that, and for users who are knowledgeable enough to dig into the raw data, we direct them to the SCEA where they can do some more complex analysis.
Yeah, excellent, excellent. Okay, if there are any more questions, we can go to the save them for the end of the workshop. So let's move on. Our next speaker is Oliver He of the University of Michigan, who's going to present on CellCards, an ontology-based knowledge base for categorization and analysis of individual cells and cell connectomes.
Okay, can you hear me?
Yes.
Okay, great. Yeah, I'm glad to have the chance to talk to here, about our CellCards project. So the CellCards is an ontology-based knowledge base for categorization and analysis of individual cells and cell connectomes. So here we have many authors of the study, including many people from Michigan, like me and my students, Fatima, Andrew, Yijia, Yichao, Michael, and we have many collaborators from 7 other universities, like Bill, Alex, Chris, Jiang, Google, Melissa, Michelle, James, Jennifer, Naki, and Sanjay. Okay, so next, first of all, the topic or the challenge we want to face is that trillions of cells in human bodies are interconnected. They are together to perform various functions, right? So for a typical human body, we have about 80 organs like here, and we have about 37 trillion human cells. And then a cell holds about 42 million protein molecules. So that's a huge system. So how to represent them in a nice connected way? Well, one thing which is more challenging for us is the so-called connectome. So connectome is basically the idea of originally describing neuron connections in the brain. So you can see so many like neuron cells. They look like a wiring diagram, but they perform very well. But if something wrong, definitely we cannot think clearly. So the connectome idea then can also be extended to other organs, right? Like we can have two types of connectomes. So one is physically cells are connected to each other and form some functional unit, like the renal capsule connectome. Another type is so-called indirect, right? They connected through messenger molecules, like the REST system. So here is more example. This is the renal capsule cell connectome. So this has the function of blood filtration. And then it has at least 12 cells interconnected. So this is the figure provided by the KPMP, the Kidney Precision Medicine Project. So you can see this is more like composed of a knot of capillary cells. So this knot is called glomerulus. And then it's surrounded by double-walled capsule. It's called Bowman's capsule, and that opens into a tubule. It's quite complicated, but they have definitely done a wonderful function for the blood filtration. So how to represent such a complex system in a relatively simple but intuitive way and computer processable? So it's challenging. Another example is the REST system, right? So this is also associated with the renal, but it can go beyond the kidney. It can involve with lungs, with blood and other systems. So it's also quite complicated. So we have two examples. And then, so we are developing some CellCards web applications. Of course, the idea actually is not our kind of original idea. The idea has been floating there for many years. So for example, by organizations like Brain Initiative, Cell Sensors Network, and then Allen Brain Atlas Initiative, and in many meetings, like a previous joint HubMap HCA meeting, they were also talking about these kind of ideas. And there are publications. This is the most recent publication, generally this year. So it's focused on the long map. They also call it CellCards. So then, what is our idea? Well, our idea is more like incorporate the previous ideas, and we also kind of thinking about the gene cards. Right? They are gene card. We're kind of thinking about a gene card idea. So like having individual pages, like one page for one card. So the card can be two types. One is a cell type card or cell connectome card, right? So cell type card is for individual cell type. Cell connectome card is a cell connectome. And then, in each card, right, you want to include more than just the type itself, right? You want to include many other topics that I will show later. And then you want to be ontology focused, right? And that fits into our sales ontology workshop here, because ontology is so wonderful and it can interlink all the things. They're all knowledge. So, but we want to bring all the different ontologies related to the cell type or the cell connectomes all together, so to make them more like seamlessly interconnected. So that's our idea, and we start with, but we like to go beyond the kidney. So this is the website. So this is our general design. As I mentioned, there are different topics, right? Like the cell type hierarchy, location, connectome. So one cell, like podocyte, can be part of the connectome. And then biological processes, it can be gene ontology, biomarkers, can be OGG, like genes and genomes, and the protein ontology, and other, and the CABI, yeah. And the gene expression, sequencing, imaging, cell line. Cell line can be the cell lines that have been developed to study podocyte, right? So this is our original design before we went to the more like a database development. So it's the original one. We designed two like more like HTML kind of format, and then we see we want to develop things like this and want to put it into more like automatic than just a manual. So we have been focused on manual collection, but it's definitely very slow. And also another focus is the HubMap ASCT plus B tables. So the AS means anatomic structure, right? They're typically represented by Arbor. And then cell types, they are more like a cell type ontology. So they're connected like it's you can hear right from the previous two talks. So the cell types are like parts of the structure. The structure can be different level. They usually call it AS1, AS2, AS3, AS4, like this. And then each cell type can have different biomarkers. So this is a wonderful story. Like you can have the information. It's kind of Excel kind of sheet or it's a spreadsheet. So we can download it from the HubMap website, and then so we can input it into our data. So actually, I'm one of the three, like me and Sanjay and Todd. We are kind of authors for the kidney ASCT table of HubMap. So we know the topic very well. Also, okay, so this is kind of once we have the tables and we have our manual collected one. So we run some program to we have used the PHP spreadsheet and some other methods, and then we run our ETL to load the data into MySQL database, and then we have a display like a PHP HTML program display results. So I'm going to show you some results we have made. So this is a quick demo. I can probably do it on-site like a real demo. But anyway, you can type a word, right? So and then you can find a lot of terms, and then you can click one and you find some information. Wow, yeah, some imaging here, some hierarchy, ontology, cell ontology. This is the ASCT Arbor terms. So you can look at this one. You go to the dedicated website. Here it is ontology demonstration of the hierarchy. And then you can also go to the Arbor, like a designated site in OS, right? So you can see all this. I will later provide some on-site live demo later. And then another CellCard demo is like this one. So this is again from the ASCT plus B. We have passed it successfully. So you can go there, and then one thing, this is a very early development. We just got the feature done actually a couple of weeks ago. So we are planning, I think we have the capability to do so kind of with the ID, right? So you can do a Sparkle query like ontology, or you can use some script, right, to automatically extract the definition and surname and many other things if we want, like a hierarchy from the ontology side to here. It's some like ongoing thing we are doing now. And then we have more like this is more like our model certain. So we have like the lineage, right? And it's a cell line cell. Again, a lot of cell line cells from cell line ontology. They have been used, derived from podocyte, right? And then you have clinical significance. Like actually, if you look for HP, human phenotype ontology, if you search a podocyte, you can see normal podocyte cell morphology. There's a list of terms that are associated with the phenotype. So actually, that's a wonderful demonstration to show there is a need, right, to integrate not only cell type alone, right, but integrate many other things, and also like gene ontology, we can look for other things, right? Many things we can do. And then this is the CellCard schema is based on the link and mail that Bill, William Duncan Bill, and Chris Mongo from Berkeley. And then we have developed the schema which actually map to the CellCard demonstration. It can be used in many ways. For example, it can be used for the data validation. So you can say, oh yeah, the ontology should be like this kind of format. If you do something wrong, they will say, oh, it's not the format. So you can say, oh, the annotation is wrong. You have to regenerate to do some collection, right? So and basically, we use link and mail and generated a JSON format, right? And for data validation. So Bill has given some demonstration. It's working fine. Yeah. So let me maybe before I go to the summary, if I can have some time, I can have a quick demonstration. So this is the website. So you can type anything basically. For example, you can type for kidney, you can type anything. This is product site. So you have this one, and you have, yeah, we hard coded. We should do it later, but this one is automatically loaded from the database. So yeah, still some part is still hard coded before, just copied from the HTML version. But if let's see if you can look for others, for example, you can. This is all automatic. There's no line of hard coding. So basically, you can go to this cell one. Again, we think we can extract the information dynamically, and this is ASCT. This is B, right? B is biomarkers. Yeah, we need to do some better job. But anyway, you can have links to other site, right? And we have a program. I think we haven't done it yet. So with the department ID, we can automatically get the full citation information and put in here. So again, it's a very early stage development, but we are working on it quickly. So in summary, the CellCards.org is an ontology-supported knowledge base that represents and stores individual cell cards or cell type cards or cell connectome cards. So we have a built-in rich database. We are getting some, we hope it is user-friendly web interface. We have a search function, a link and mail schema. So again, we are still at its early development stage with many live pages constantly updated. So currently we have kidney about 70 cells already there. So we are now adding more data. I think lung will be the next one, but then we want to add more than 10 other organs data. So eventually it can be hundreds of cell types quickly. But then we need to look for other resources. Like our literature, our manual collection is quite slow. So we probably need to look for other resources and integrate them together. And we can also look for some KPMP or HubMap, right? We're trying to get some collaboration with them. Okay, so I think that's it. Yeah, so yeah, basically here we have some wonderful undergraduate students like Fatima, many students. And you are here. So and we have people from like Bill Duncan, initially in Berkeley and now in Florida, Pinaki and Alexander Chris Jiang-Guk on working on the literature mining, Melissa from working on the imaging. And Cindy is our domain expert in kidney. Okay, and we also like to support to acknowledge KPMP and HubMap. Okay, that's all I have. Happy to answer any questions. Thank you.
Thank you very much, Oliver. I'm sure there might be some interest in this. It overlaps with some other related projects. Would anyone like to ask a question? So Oliver, obviously lung map is doing something similar, and Clearly, it'd be nice to collaborate, but also, I mean, I can see how some of the development of the interface could be also inspired by, what FlyBase is doing with their cell type data, and perhaps some other databases as well.
Yeah, that's a very good point. Yeah, happy to learn or collaborate with the Fly base project. Yeah, it's wonderful talks. Yeah.
I'm wondering about, so all of the data is downloadable in LinkML. Is that the case?
Yeah, that is the plan. I think Bill. Yeah, Bill Duncan, right? Bill has generated couple, right? It's only a couple of cells there. We haven't done it. Was originally like put together, as some HAMAP submission, grant submission. Yeah, we didn't get it.
Oh, yeah.
But then we think, anyway, right? It's this is one of the 3 or 4 aims, but we think, we don't get others, but we should make sure the CellCards project should go forward. So we have put in a lot of effort here now.
Yeah, I mean, I think, clearly there's going to be lots of different efforts, building these sort of knowledge base, CellCard, Atlas type resources, and the more that we can sort of easily share data between them, I think the better. It's obviously going to be a challenge, but especially, I guess a lot of it, what you're doing is integrative. It takes existing links between ontologies, but then I'm interested in the accessibility of the novel data there as well. So the connectomic data, is that some stuff that's available directly in KPMP, or is that?
KPMP has many good projects, many good data. Yeah, I'm a guy of KPMP, so I've been talking with them. So David, I think I really like your talk. Wonderful. Maybe we should talk sometime later and see if we can have some kind of collaboration. Yeah.
So I just want to say, like the purpose of the LinkML, right, is there's lots of ways to accomplish what it does, but one of the main things is to provide an easy way to make the schema public, have documentation, and also produce different validators like JSON schema or Shex or Shackle, so that way we can keep the schema and the validators in sync along with the documentation. And then of course, if you have data you'd like to submit, then of course we have at least an upfront way to validate it.
Yeah,
like in terms of the
sense.
schema.
I'm definitely interested in LinkML for some projects for that
Yeah.
usage. I suppose I was interested in it also for download. So how, does this give me standard? I guess the underlying database is MySQL. I don't want to deal with a MySQL dump, but if I get something really standardized.
Yeah. So the download, right? There have to be a layer that say takes the MySQL, right, and converts it into JSON, right? That conforms to the schema. That's probably the best path there.
So that doesn't, right? So right now it's for loading into the ETL pipeline. It's not two-way.
It could be if the backend, say, was a Mongo document store, which we've done in another project, but right now, because the backends are, is a relational database. Yeah, there has to be some kind of version layer.
Thanks.
Great, thank you.
All right, thank you very much. So our next talk would be by Joshua Fortriede, and on the rapid prototype for cell type data catalog collection interface. So, thank you very much.
Thank you, Alex. As Alex said, my name is Joshua Fortriede. I work on the lung map project. So Oliver, if you want to reach out, more than happy to discuss things with you. I think there's a lot of overlap with what we're doing. And as Alex said, my long mouthful of this. So we've been working on this, a method to catalog all of the cell type data that we're working with. And we're in the very early stages of this, and so trying a system that allows for this sort of rapid prototyping of this collection interface, so we can add features, add fields, remove fields that we don't think that we need, to be able to easily generate reports and stuff. And so, what's the initial problem that we're trying to solve? As everyone on the call knows, there's been a rapid expansion in single cell omics and multi-omic data sets. We have all these specialized groups looking at organ or tissue level spaces. We heard about the HubMap ASCT plus B tables, the LungMap CellRef or CellCards. There's the human lung cell atlas. And so there's a lot of these. And with all these initiatives, they're identifying very specific cell types, or even in some cases, subtypes or cell states. And so this is this is really growing the amount of data that we have on very, very unique and specific cell types. And note I say data and not knowledge, because first we collect data, and then we have to look through it and filter through it to be able to get that sort of knowledge out of it. And I found this graphic online, kind of showing where the field has gone. First we had all of this bulk data, bulk RNA-seq, and that's more like the fruit smoothies that you can get. And then we have all the single cell data, which is all of the individual fruits that go into that smoothie. And then we have all this spatial data, where those fruits are, and in what format are they? Are they sliced? Are they diced? Are they whole? Are they fresh? Are they frozen? And so there really is a huge amount of data that we're trying to work with, and to be able to work with it, we need to be able to collect it in an easy to use system, so that we have it cataloged, and people can gain access to it. And so the solution is a sort of cell type data catalog. And when I first thought about this, I was inspired by those Go annotation tables that a lot of people use. So you submit Go annotations of this gene is associated with this biological process, and it's in this species, and here's the publication for it. And there might be a few other fields, but for the most part, that was it. But you generate huge tables of these, and that knowledge was used in a lot of places. And so that's kind of what inspired this idea. And so again, the goal was to be able to create this sort of curation interface for the curation of cell types. And we wanted to be able to collect a wide range of metadata, including biomarkers. I think everyone on this call believes that the biomarkers is probably the most known about a lot of these cell types, specifically coming out of single cell genomics, but also to collect the cell descriptions, functions, morphology, synonyms we've heard about, these anatomical locations, because maybe a cell type in one part of the body is actually different from basically the same cell type in another part of the body. We're collecting disease and phenotype information, and then even cell state. And so as we were designing this and thinking this through, we had a couple of requirements. The first, we wanted to use ontologies to accurately and uniquely curate the metadata. And I'll show you several of the ontologies that we use. But then we also wanted to compare similar entries to distinguish like duplicate cell annotations in our system versus subtypes. And we'll have examples of those. We wanted to be able to easily generate summary reports. Again, getting data, collecting it in a system is only good if you can pull that data back out easily. And then, we wanted to be able to easily extract that data for other initiatives. CellCards, now, when I wrote this, it was the lung map CellCards, but it could easily go to the other CellCards or to ASCT plus B tables. And so one word of note here is the purpose of this system is not to be a definitive final answer of a cell. It's not to say, this cell has this definition and these are the markers, but it's a collection of data that we can use and combine together and validate together to establish that answer. We heard in a previous talk that the cell types that you get from one data set and the marker genes may not give you the full answer of marker genes for that specific type of cell. Maybe it's because they were treated a certain way, maybe it was a technology, maybe it's the specific subtissues within an organ. And so this work is to be a step before that, just to collect a broad amount of data so it can be analyzed. And so to do this, we did all this prototyping using REDCap. And REDCap is used a lot in research-based clinical trials and studies. It's very easy to be able to add forms and add reports and stuff to it. And so that's what some of these features are. So with REDCap, you can quickly build these data entry forms. And these forms, you can specify fields and give certain types to fields. These are number fields, these are text fields, we have drop downs. And one of the most important things is in REDCap, you can have ontology term fields. And these are type aheads that utilize the BioPortal API. And so we've been able to easily incorporate several ontologies into these forms. As I mentioned before, one of our requirements was to be able to build and generate reports. And REDCap allows you to very quickly and easily do that. And then finally, REDCap has a robust logging history and comments features. And so you can see who put in what information, when, you can add comments. And so if you can't quite get the answer into the fields we have, you can put a comment on the field so others can see it. And so right now I'm going to go through several of the screenshots of this system that we've put in place. So when you first go to enter a new type of cell, this is a form you get, and here we have it filled out. And so we're collecting information such as species, and then at the top, what do you want to call this cell? So here we want to call them, capillary 1 cells. We then get into the hierarchy or levels of the cell. So we know that these are endothelial cells, and then we start to put in some terms from the cell ontology. Well, we know that this is a lung endothelial cell. We know it's a pulmonary capillary endothelial cell. We know it's a lung microvascular endothelial cell. And so this is going to help us later on try to take this information and put it into things like ASCT plus B tables or CellCards, or give us information for where to put new cell types in the cell ontology. We collect cell state information, and so there's several checkboxes here that can be checked. You can specify specific anatomical terms. I'll show you some examples later where that comes in handy. Several times we include these sort of free text note fields where you can just put a lot of information as well if it doesn't fully fit in a specific term. And then one of the final things we collect here is, if you were to guess, what is the closest cell ontology term to the cell that you were trying to describe? Now, in this case, we have an alveolar capillary type 1 endothelial cell as a cell term. And then the relationship of this cell to the cell ontology term is my cell is exactly the same term. I know this because I created that cell ontology term. And so this is trying to get the submitters to document this and start to think about this as they're trying to fill it out. After we've collected, yes, David?
Yeah, I'm wondering, sorry if I could, I'm wondering about, so the restrictions you put on this for a start. So superclass seems to be an Uberon term rather than a cell
Yeah,
term. So you could have endothelial cell there, or?
yes. So this is still prototyping, and I would
Okay.
definitely write that down as
And,
something to change.
and I guess I wanted the sense of what the levels were for. So is this to deal with multiple inheritance classifications? So you might, because because I would think pulmonary capillary endothelial cell probably already is a lung endothelial cell in CL.
Correct.
I mean, I suppose there's no harm in redundancy, but.
And so as I've worked with non-ontologists with dealing with ontologies, it's for a lot of researchers, it's very hard to use ontologies. And so what we're trying to do here is just collect some information from the submitter. You know, and then later a more senior curator can go through this and go, okay, I understand what the submitter was trying to get at. So it's not trying to be a definitive lineage. It's trying to just be a guidepost for future curators.
Yeah, it makes sense, and I think we really need to do experiments with users to find out what are the most efficient ways to gather this information. Okay, thank you.
Yep. And so after that, we start to collect some more descriptive information about the cell. Synonyms, you know, you heard David and others talk about this before. You need to be able to collect synonyms because, you know, everyone who likes to name a cell likes to name it their own thing, and then they like to give a short synonym for it so that they don't have to keep typing it throughout their entire paper, and it's easier in figures. You can also provide DOIs. So if another paper has mentioned this, then you can give a DOI to that sort of paper. Then we have fields for sorts of descriptions, morphological features, any sort of cell function, developmental origin, regenerative potential, and so on. And this is very helpful, you know, because it helps identify a cell. What's, you know, what is it? What is its, you know, features, and it allows other people to know, yes, that is the cell that I'm talking about. Now we're getting to the meat of the data. And so this is primarily, this is the area where most of the data will be filled out, because a lot of these are coming from single cell genomic data sets. And so what we're doing here, again, is collecting these gene biomarkers. And so you can specify up here up to 5 different data sets. And so you give a short data set name, and then you can give DOIs, URLs, PubMed, citations to it. After you've filled out those data sets, now you can come down to this box and you can start putting in genes. For these gene boxes, we are using the Human Gene Nomenclature Committee. They have a ontology and bio portal, and so these are lookups, so you can type it in. You click on the term and you get an ID, which is very vital for this type of work. And then you can have checkboxes to say, okay, I see this gene as a biomarker in these data sets. And then if you can't find a gene, you can put that sort of information down here. Similar to genes, we also collect protein biomarkers. Again, this is very similar. You specify your proteomic data sets at the top. Then you can specify the specific proteins that you're looking at. You can specify types. Currently, we only have a surface protein. You know, that's important for biomarkers, because you need to be able to know if you can detect a cell by a surface protein. And then you can specify what data sets define those proteins as biomarkers. And then one thing we have here that we don't have in the genomic biomarkers is imaging evidence. And so lots of times there's imaging evidence of these proteins. And so down here, you can specify the protein. This is going to be a drop down based on what you input up here. You can specify the data set. Again, this is a drop down based on what you have up here. You enter in a URL, you enter in what color or channel that protein is in the image, and then you can put in an antibody RRID, and we have a link out to the RRID website. So if you don't know it, you can go and look it up. And again, we have the sort of free text area down here just to put information if you can't find those biomarkers, or you have more than 5, or there's something else you want to specify. The next thing we collect is disease and phenotypic information. For a disease, a user can pick between 3 different ontologies: the Mondo disease ontology, the human disease ontology, and PED terms, just because the lung map group has a lot of pediatric samples, and so the PED terms is very helpful for that. And you can specify the ontology term. You can give any sort of notes, pathology, or other findings. And then if there is a good image of the cell type in the disease, you can put a URL in the image box here. We also collect the phenotypic information. Here we just limit it to the human phenotype ontology. And again, you can specify notes as well as an image for that phenotype. So with all of these data entry fields, there's also 2 extra buttons. There's this little button here that has an H in it, and then there's this speech button. And so what those do is the H button gives you the field history. And so you can quickly see, okay, who entered this information, what information did they enter, when did they enter it? And this is very helpful just to track the provenance of all of this. The other thing is we have the field comments, which allows you to put in comments. If you're not quite sure about the data going into a field, you can put the information in the comments. Here I state that the paper states that this is CAR4. NCBI gene shows that CAR4 and CA4 as the same gene, so that's why it's CA4. Just it's a little note to myself as to why it says CA4 and not CAR4. And so what this looks like in REDCap is we have these different records for the different superclasses, and then when you go into one of these, you can see all of these different annotations or cell types. And so here we specify the name that was given by the submitter as well as the species. And so what we can do with all this data is we can quickly generate these sorts of reports. So here's a report of all of the different cell types, the species, and then the synonyms that were given and the closest cell ontology term. So you can quickly browse and find terms based on synonyms. We can generate gene reports, which is very helpful for several reasons. The first reason is finding biomarker differences across species. And so here, if we look at capillary 1 cells between human and mouse, we see that there were different gene biomarkers established, as well as protein markers. Same for capillary 2 cells. In this example at the bottom, we can see biomarker differences across cell states. And so here's alveolar type 2 cells in human and mouse. They have the same biomarkers. But if you look at a different data set that found alveolar type 2 cells that were proliferating, they have different genomic biomarkers. And then finally, we can quickly identify biomarker differences across anatomical regions. And so here we have serous cells in human and mouse. Again, same genomic biomarkers, but if you separate this out and you're looking at bronchial versus nasal tissues, now you start to see different biomarkers depending on the anatomical structure where these came from. We can also quickly display all of the diseases and phenotypes. So we can have a quick table that we can browse to identify diseases and cell types that are associated with those diseases. And so the next steps are we're working on beta testing. So there is this Lung Summit group that we are working with to beta test this. That should give us vital information on how to expand this. We're working on building more reports just to get more views of the data. One thing the RegCap allows is these sort of access controlled forums. And so that will allow a certain group of users this sort of higher level curation or specific curations. And then finally, we want to work on exports for integration with other initiatives. Again, ASCT plus B tables, CellCards, etc. That's all I have. Are there any questions?
Thank you very much, Joshua. It looks like a very comprehensive system. I think Tiago had some comments about his work on the Miracle standard. And
yes, that's another one of the things that we would want to build a report for. Is we could easily build a report that is well aligned with the Miracle standard, and that would allow for your rapid integration into the cell ontology.
Yeah, I have a question here. So it seems like, yeah, there's some overlap between the long map and the ASCT plus B, right? But I also know ASCT plus B is more like for the healthy cell, like a cell markers, healthy cell markers. So it's like your long map focused on like a diseased cells or diseases, yeah.
That's a good question. So yes, you are correct that the ASCT plus B tables are more focused on healthy on healthy cells, and that's really where you need to start, because you need to describe the state of healthy cells before you can describe the state of diseased cells, right? You need something to compare to. Similar with lung map, we are focusing primarily on healthy cells, but and specifically with lung and lung map, we're in an environment right now where we have a respiratory, disease that is spreading. And so having the ability to also focus on diseased or infected cells is vital. And as we're building this and as we're prototyping, we want to think about the future, because again, we went from bulk RNA-seq to single cell RNA-seq to spatial and cell states. And one of the next steps is going to be disease. We know that. So building the infrastructure for it now helps us for the future.
Thank you.
very interesting. So do you think you're going to be adding or using this system as a way to find many new cell types for inclusion in the cell ontology?
It is quite possible. As I sat on some of the cell ontology talks, one thing I know is, there's debate on how to add cell types to the cell ontology, or what evidence do you need, and are cell states, consider different cell types, and so the reason for doing this is to collect that sort of information, so we could utilize it to add terms to the cell ontology. And we realize that there are restrictions to add terms to cell ontology, and there need to be. And so if some of the cell types can't go into cell ontology, maybe it goes somewhere else, or at least it's in a database that can be utilized by other people.
Yes, thank you. That sounds like something we'll have to work on in the future.
Yeah, I think bearing in mind the things as candidates for the provisional cell ontology there as well. So thinking about some kind of export or linking to pull stuff together could be really useful.
And I mean, this system is very easy to export data. There's APIs to be able to grab data and do stuff with it. So being able to get information out of it for these other systems is easy. It's just what is the quality of the information that people are putting in here? You know, with ASCT plus B tables, you know, those have curators looking over them, and there's things, you know, there's scripts and pipelines to synthesize it and look at stuff. So there's always a cleaning aspect of data, right?
Yeah, but I think thinking about building pipelines of potential new terms for CL and PCL, I mean, depending on resources as well, but you know, trying to pull some resources to push things down that pipeline could be useful as well.
Yeah, I was going to say years ago, I did a lot of work converting REDCap into RDF. If that would be helpful. It was on an earlier version of REDCap than what you're probably using, but I imagine the underlying schemas haven't changed that substantially.
Yeah, I mean, that does sound interesting. I know that you can export in multiple formats. I don't recall if RDF is a format that they support now, but that would be interesting to look at.
Okay, I think we're going to have to move on, though. Getting a little behind schedule here. Thank you very much, Joshua. And I'd like to introduce Patrick Ray, who's a longtime friend and collaborator, another product of the University at Buffalo philosophy department. And Patrick, please talk about your work.
All right, I can't see my screen.
We can see your first slide.
Okay, just the first slide.
Well.
Okay, good. So thanks Alex for hosting, and thanks to all the presenters so far. It's been very interesting to hear all the work that's been done, and I'm going to talk a little bit about some other project. So I work at the Allen Institute, and we have a lot of different projects, of course. I'm sure you're familiar with. David talked about some of them, and it seems like other people are aware of a lot of the work that we do. We have a cell types knowledge explorer. Of course, we have anatomy atlases for various types of various species of brains, and we're kind of expanding on this work to characterize all the cells in the brain and do things like that. So lots of exciting work. But today I want to talk about a project that I was involved with and still involved with that has to do with electrophysiology modeling at the cellular level. So I'm going to go through some motivation and some goals and a little bit of background on this. And I'm not going to be talking directly about cells themselves in this talk, but I'm going to be talking about the experiments that we use to generate data about cells. So we have most of the talk so far have been about all the different data. Most of it's single cell or a single nucleus or just some sort of sequencing data. And there are lots of different experimental protocols. There's an explosion of them. It seems like every day there's a new one, and we have to figure out what is going on in these. And it's really important, I think, if we want to characterize the cells, that we understand what's going on in the experiments as well. So the E in the cells is kind of what I'm interested in here. And so I'm going to talk a little bit about patch-seq today, but of course, this is just one type of experimental method that we can use. And so in patch-seq, and again, I think David covered a little bit of this, but what you do is you basically, you can have slices, or you can have cultured neurons, or you can have it even in vivo here. And what you do is you take a pipette and you put it on a cell body, and then you apply a little bit of vacuum to it, and you can inject a current into it, or you can clamp it somehow. And then you can take a recording and you can get data as a response to that. And then you can also get morphology data. And then you can sequence that cell. So we can know a lot about a single cell. We can know about its electrophysiology, and we can know about its morphology. And then we can also determine its transcriptome. So in this case, I have a kind of little diagram here of the different ways that you can do this on the left. And then on the right, this is a little figure that I took off our cell types knowledge explorer. So this is one of the I guess data modalities that we use to annotate cells on our cell types knowledge explorer. And so you can see that these are traces for an individual cell, for an individual cell type, and in response to some injection current, different types of things. And so these are data that we want to have for especially neuronal cell types, and we want to understand what's going on in the experiment that generates the data so that we can annotate the cells with the data. And so basically what we want to do is we want to, if we want to characterize these cells, is we want to be able to characterize everything that's going on. And so the goal of this project was to kind of start at the beginning. So we're starting at the experimental level. And so a lot of times when you look at an experimental protocol like patch-seq, you have these pulse waveforms. Here's a really simple one, the most simple one that we can have, which is a rectangular pulse waveform. And so you just have some time T1, and then you inject a current or a voltage or something like that. It's different ways that you can clamp. And then it goes back to base at T2, and you can kind of string these together so you can get double pulses, things like that. And then if we kind of look at the diagram on the right, we have step transitions and trapezoidals and step transitions down. And then we can kind of string these things together too, and these like double pulse, and then we can have a bipolar pulse, we can have a staircase, and we can get compound waveforms, and it all gets very complex very quickly. One of the things that we wanted to do was kind of standardize these, right? So we looked at all this patch-seq data, and we said labs here produce this data, and they use and reuse these stimuli, patterns, or types. So ramps and chirps and steps and experiments. And if we want to be able to compare cells, or if we want to be able to take some experimental data and say definitively that it is a certain cell type, we have to know exactly what is the input for these patch-seq experiments. And so we decided to start there, and then we were going to expand further into the responses that we get. And so we're going to create an ontology, basically, for the stimulation patterns that are inputs to these patch sequencing experiments. And of course, there wasn't a universal standard for these. Sometimes neuroscience can seem kind of like the Wild West, people doing all sorts of different things in different labs all over the place. And so, it's just notoriously difficult to get people to agree on this, especially it seems like in electrophysiology experiments. And so, what we have when we look at the landscape here is that each researcher is kind of in this position where they have to determine for themselves how to describe the stimuli and what metadata need to be included in this description. And so we had a working group, basically, and the working group kind of identified some further problems. And so we don't have any standardized description and metadata requirements. And what happens is that we get frustrated researchers and frustrated data curators and ontologists that are trying to identify similar protocols across data sets. And when we want to do data analysis, which is kind of like the end point for a lot of this work that we do, it's really hard to do this if we don't have any sort of standards. And this is troubling, especially in the era when we're talking about big data, which at the Allen Institute we're very committed to, and FAIR. Again, I think we're all committed to FAIR as well. And so there's some effort here that we can standardize this type of data and data annotation, and this can help laboratories and researchers. So we don't only want to create kind of end user products, but we want to get in touch with the labs and the researchers and kind of help them as they generate data, or perhaps before they generate data, so that they can have a standardized way of talking about what they're doing in the lab, so it's easier for us to annotate these cells down the road. And so we need to expand the tools that are available for the annotation of this data. And there's often a type of redundancy in the stimulus set that defines a single experimental protocol. And that's because the stimuli typically differ by only a single parameter. So for example, amplitude, latency, or frequency, and that's varied over some range of values that we have. And so there is a way to do this, and it's kind of all very close. So it's for those of you, I guess all of us who have worked on ontology for a while, it seems like the problem is very close to being solvable, except there's kind of one little thing, one little wrinkle that kind of prevents it. And so while everybody seems to be doing the same thing, it's not super standardized. In fact, it's not standardized at all. And these little values kind of change. And so we need to figure out a way to talk about these types of things. And so those variable parameters need to be made explicit when we're talking about the stimulus representation. And this would allow the stimuli to be represented compactly. And this is basically a necessity for more complex types. That's not just a 1D time series for experimental protocols. And so this not only applies to patch clamp experiments or patch sequencing experiments of which patch clamp is a step, but other one-dimensional time varying stimulus types. So extracellular stimulation and optogenetic stimulation as well. And there's not any ontologies that seeks to standardize any of this. And so we went ahead and built a small application ontology, which is called the electrophysiological stimulation ontology, which is a mouthful. So what we're going to do is we're going to define a canonical set of one-dimensional stimulus waveforms that are used in intracellular ePhys experiments. And the parameters that are needed to describe each stimulus type. Of course, we're aiming for the minimum. As David says, typically the minimum is what you want in this case. And then we want to develop representation in NWB, which is Neuro Data Without Borders, that supports parameterized stimulus definition and lets us see the variable parameters for search and that can reveal experimental design. And so we set out to determine a set of stimulus metadata that's going to be defined at each level of experimental protocol. So sweep, sequence, stim set, condition, etc. For our experimental descriptions. And for their output, the team is to design some visualizations for stimuli and stimulus protocols. So the first thing we had to do was kind of go through and look at what people are using for waveform generation and kind of try to conform to that. So considerations, these are some of the things that we're going to consider. I'm going to run out of time, so I'm going to skip this slide quickly. Here at the Allen Institute, we use something called a MEES wave builder. And the MEES wave builder provides a set of atomic waveform parts that you can combine into a stimulus waveform. And the wave builder module is used to build what we call stim sets in MEES. And the wave builder is used to create sweeps. And we call these sweeps a stimulus set. So sweep can be composed of up to 99 epochs, which is quite a few. But if you've done this work, you know that you want to string some of these together very often. And each epoch can contain some, preset number of waveform types, but it can also contain, you can also kind of adjust it so you can have arbitrary combinations of waveform types, and you can also just customize a waveform. So, again, start off with something that seems pretty easy. We just have 7, and then we have arbitrary combinations, and then all of a sudden you can do anything you want, which is an ontologist nightmare. And you can kind of save your custom configurations, but you can also have custom configurations or waveform configurations that can be a sweep from a different stimulus set or something that's generated from tools outside of me. So here's what the wave builder kind of looks like. Here are the parameters that we're kind of looking at when we open this up. And you can see we have kind of like tabs on the top here that with your kind of 7 waveforms generally, but you can also, increase it in the epochs and the sweeps. And you can save it and kind of load things externally, and then you get a kind of nice, you know, you can change the time and stuff on this. It's actually pretty easy to use, I think. And, you know, at the Institute, we have a kind of standardization of these for PatchSeq experiments. And so this is kind of from our handbook of how to do these. But you can see this is kind of a summary of the different basic types of waveforms that we use here. And so we look at these stimulus segments. There are some parameters that are defined for all segments. So this is information that we want to collect for all the different types of waveforms. So things like start time, and this is relative to the start of a sweep, duration, of course, offset and baseline, which are kind of diagrammed here on the picture. And then we can also have stimulus segments. So these are the kind of the tabs when you look at the MEAS. So we have square pulse, of course, and you know, we're mostly concerned with amplitude, but of course we can extend these in time. We have pulse trains, which is just a train of square pulses. And those things, you have amplitude and duration, and then frequency and number of pulses. We have ramps, or what are called sawtooth waves, if you kind of take ramps and string them together temporally. And here we're dealing with a slope on this one, so slightly different. And then we have what are called chirps, or sine or cosine waves. And so chirps are, as you can see on the diagram here, they can get very complex. They're kind of easy to describe mathematically, but much harder to describe ontologically. We have other things that are really helpful in experiments, like noise. So we have different types of noise. We have white, pink, and brown noise that we use. These also have cutoff filters. And different filter parameters that can be associated with those segments. And then maybe the one that might look most familiar to us is a PSC, or a postsynaptic current. And so we have a PSC waveform as well. So so when we kind of sit down and look at this, these are things that we want to consider. And so remember that these can be combined into arbitrary waveforms, and you can also kind of stack them on top of one another additionally, and so you can have more than one kind of going at once, but you can also concatenate them kind of temporally as well. And again, you can also have custom waveforms. So when we started doing this, it gets pretty complex. So we have a set, so I'm gonna, let's see how much time. So here's a bunch of stim sets that we have defined. I'm going to skip through these because I'm running out of time, because I've talked about them too. But we also have data from integrating data from outside the Allen Institute. So things like the Blue Brain project here, where they have a set of stimulus functions also defined. And so a lot of them look the same. So we have AP waveform kind of here in orange, which looks a lot like a square pulse. And then we can have these kind of like step square concatenations as well. This looks like a noise, and of course this looks like a sine, and these are kind of like ramps. So a lot of these look like things that we already have defined, but they're just slight variations on it. And so you can see the problem, I think, kind of manifesting when we start looking at some of this other data to integrate. And so another thing that we were in the course of the project is we're looking at experimental methods, and we want to be able to annotate these papers, or at least descriptions of what's going on in the experiment for sentences like these. So a sequence of single monophasic current pulses, duration 1 millisecond, was delivered at 0.1 hertz at 4 intensities organized in randomized blocks of stimuli, 40, 60, 80, 100 microamps. And so so for a sentence like that, we'd want to be able to describe that in the ontology, or at least have classes that are going to be able to annotate data like that. We have these a lot, and so there's all sorts of different parameters that we have in the descriptions of what's going on in these experiments. So just examples of things that you find, obviously. So challenges to doing this, I think that we can all kind of pick up there are multiple ways to specify the same waveform. And sometimes we're just going to have to pick a convention. So there's not going to be an obvious kind of standard that we all fall in and agree to. You just have to pick something and do that. Another challenge is authoring. And so authoring using semantic representations is not as expressive typically as simply composing the base waveforms. And so somebody working in a lab that's using these can easily plug in all the parameters that they need. And maybe we don't have every single possible type of waveform described in the ontology, which would be a ton of things to describe. And so a lot of times they're just going to build their own and not have anything that's corresponding to the semantic representation. And so what this means is that authors will sometimes fail in kind of making these things reproducible. So one of the solutions that we discussed in this working group was to allow the waveform authors to tag non-semantically authored waveforms with preferred nomenclature. So that, if they wanted to make their own waveforms, that they can just kind of create their own tag for that too. And so in that case, we would want to have some sort of tooling that writes detectors or definitions for things like pulse trains and bipolar pulses and things like that. As we started working on this, the general requirements we wanted to define different types of waveforms, including states and transitions, and try to get this to conform kind of generally to descriptions that we found, in particular here in IEEE document. We wanted to kind of group functions by family. We wanted to describe function parameters, so period, frequency, wavelength, amplitude, and offset, things like that. Other things that we want a stimulation target, so this cell body or presynaptic cell body or fiber tract or various other places that you can put electrodes. Also, if we're going to allow people to kind of make their own waveforms, we need things like addition, concatenation, function composition, and we need to define kind of precisely what those things mean. And we need to also include things like effector modality, so electrical charge, magnetic field, et cetera, et cetera. And so we started off kind of constructing some general terms, so stimulus and response, sweep and sequence. Again, I'm going kind of quickly here because I prepared way too much. We wanted to define kind of runs because these are kind of the basic units that experimenters discuss, conditions, and then stimulus types as well. We needed some relations. And so we created some relations in the ontology, things like reference level, duration, originating level, terminating level. These are necessary to talk about the different types of waveforms. And then we kind of set off developing the ontology. So this was a collaborative effort with a group of people coordinated through the INCF. So we have an INCF working group, and we created an ontology that kind of grew out of this. So it's called the eSTEM ontology, and it's a small ontology. So it's not too big because we wanted to be kind of general and capture all of the things that I just described. And it integrates with the NWB schema for stimulus description and defining required metadata. So we also wanted it to integrate really well so that experimenters can use it and can plug their data right into it. And so NWB is a nice standard, and we got a lot of support from them. So it was nice to work with them. And our development initially centered around use cases from intercellular electrophysiology targeting protocols. But we also define a specification for parameterizing stimulus templates as well. And we want to extend it to two-dimensional or visual stimuli, and that would be a further application of the work. Some basic kind of details of the ontology. It's built with careful consideration of Obo Foundry principles and FAIR principles, although we, I don't think we're aiming to be in the Obo Foundry, but we do like to be able to interoperate with Obo Foundry ontologies. And it's available in standard formats, OWL, Obo, JSON, and Turtle. And we try to use and reuse existing ontology classes as possible, although there aren't very many good ones and describe exactly what we want to describe. We have about 422 classes, so again, not super big. And this is just basically for the annotation of electrophysiology data. And then it was kind of co-developed, and so a public version would be at the SciCrunch NIF ontology. And so what we did is we integrated the SciCrunch NIF stimulation ontology into an eStim ontology, and then put that under BICCN. So the BICCN is a Brain Initiative Cell Census Network. But that project is ending, and we'll be on to a different project soon. So we'll probably migrate this work over to that. If you look at the ontology, so if you kind of like open up the stimulation ontology, it's kind of messy looking because a lot of this stuff is not well defined in terms of hierarchy. So when you look at the hierarchy here on the left, there's a lot of things that we need to describe. And we didn't really worry too much about placing it under, kind of structuring it under BFO so much in that case. But we did put things, as many things as we could in here, and tried to write as many comments and definitions and annotations as we possibly could, while remaining flexible to experimental protocols. What we want to do in the future is assist in the annotation of different sorts of experimental data. So we want to expand. We want to extend beyond one-dimensional waveforms. And that means going into higher dimensional waveforms, so visual stimuli. We want to align more closely with multimodal experimental data sets and techniques. So PatchSeq, of course, but any other ones, and I'm sure there'll be a new one next week. And we want to make modules that support annotation of data generated to describe cell types from experimental methods like PatchSeq. And so since it seems like everybody's doing sequencing data, this could be something that we could reach out to people, the BDSO or the PCL, and kind of see if they would be interested in using some of this, and if we could harmonize across that. So acknowledgements, INCF working group here, led by Tom. So Tom Gillespie, UC San Diego is the chair of that working group. And then Lydia, Pam, and myself from the Allen Institute, and Andrew, and Linus, and of course, Oliver was a tremendous help, and of course, Ben as well. So yeah, so I had a lot of support from them, institutions, INCF, Human Brain Project, Neurodata Without Borders, Spark, and the Allen Institute, and we had a grant for this too. So there's a grant, and of course, thanks Paul Allen, and thanks everybody at the Allen Institute. So yeah, I'll happy to take questions.
Thank you very much. Yeah, go ahead, David.
Hi, Patrick. Yeah, so I guess I'm curious about whether there are a smaller number of sort of response terms which we could, so the downstream data analysis rather than the upstream experimental conditions that we would need for sort of as a very high level characterization of electrophysiology data that we could use, to index things to make them findable. It seems like a slightly different issue to the sort of quite complex details of the initial inputs. I mean, are there some things that you could state to say that a cell is spiking, for example, irrespective of the input? And have you started to look into those kinds of?
Yeah. So the scope of this working group was specifically for the input stimulus. So we thought about what it would take to extend to the responses, of course, which is something we need to annotate cell types directly, but it just wasn't within scope. But I think, when we looked at the problems and we looked at the data, I think a lot of the things that we encountered with the protocols, so that's to say the stimulus set, are going to be the same problems that we're going to encounter with the response waveforms as well. So we're still going to be looking at amplitude and frequency and you know, time parameters and things like that to characterize them. So I think if we can get harmonized from kind of the input and the output, I think we can find a common set of terms that we can kind of align on those, even though they're going to be for different functions or different use cases.
And I know when we've had discussions in the past, like INCF meetings years ago, about standardizing this stuff, the general response has been from a lot of the people involved that the initial conditions vary so much that everyone's like inhibited to really come up with some very simple abstracted set of response terms. So maybe this is the right way around to do it. Yeah,
and that's why, you know, I think our approach was just get something out there and then let people reject it rather than you know, wait for people to agree, because if you're waiting for everybody to agree, I think you're just going to wait forever.
So can you imagine situations where classes from this ontology could be useful in defining a cell type?
Yeah, you know, I think the experimental data is interesting. I mean, one, we want to just you know, because we have so much data, we have so much access to data, and we want to kind of harmonize this data. You know, one way to do that is kind of to harmonize the things that happen to cells in experimental protocols, and so it's useful insofar as you want to do that. So if I want to look at two experiments and say, like, these two people did the exact same thing to the exact same cell type, I want to be able to know for certain that that's the case, and that was kind of our use case there. But you know, I can imagine a situation where, or at least I think this is what's going to happen, is that these cells are going to be partially defined by these functional characteristics. And in order to have a functional characteristic, you have to know the input, I think, and you also have to know what the output is. So, you know, trying to get the one part, the input part, standardize that, then maybe we can standardize the other part, and maybe through those two things and kind of put them together, we can have a better idea of what the cell is doing under certain conditions. So we'll need both the conditions under which the cell is put, and then the response the cell has under those conditions as well.
Yeah, so I mean, I think this is very nice work because it does address the aspect of how do we represent the experimental conditions and outcomes. I mean, I wonder, like, do you foresee that this could be something that could be linked to EBI in some way, in some respects?
I mean, yeah, I think it could. I mean, in principle, it could be linked to EBI. I don't know if they'd, you know, like I said, the EFIS stuff tends to be a little bit messy sometimes to deal with. But we could get a simplified set, I think, and kind of connect it to EBI. I think that would be really helpful for experimenters, and then we can kind of give them templates to fill out so that they can describe in detail their experimental protocols and the specifics of those, and that would help us with data organization and annotation on our end, for sure.
Yeah, I mean, I think that might be one way to add structure to your ontology. Sure. Okay, are there more questions for Patrick? So I just want to open up then the comments and questions for everybody who's given a talk today. And if you have questions for any of the speakers, please speak up now.
Yeah, Martin again. David, I have more questions for David. So I was very fascinated by your talk, David. And you described the provisional cell ontology and said it's data-driven, which makes a lot of sense to me in the context of single cell biology. So do I understand this right? If someone, so the cell ontology, the new cell ontology is data-driven, right? So that means, I think that annotation to the cell, this new cell ontology will also be data-driven, right? So my question, my question is how do you envision, and how do you envision, let's say I'm a researcher in Fly, right? And I analyze the brain, single cell biology. I have a new data set and I have clusters, right, in my single cell analysis. So how do I go about determining which clusters correspond to objects in the provisional cell ontologies and which clusters don't, and how would that work in a practical way? The use of ontology.
We have a couple of examples. So there's the Azimuth website has a bunch of standard sort of reference standard data sets. They've got kidney and a few others, and they use this. So Azimuth, you can upload your own data and it will automatically annotate or suggest annotations, projections based on the data, and that will come from the annotations on the standardized data sets, and they have, to a pretty decent depth, cell ontology terms, and we're working to sort of deepen the ontology terms that they have. There's a bunch of other, I think it's a matter of getting these things out there into the infrastructure. There's a very large collection of sort of reference data sets, a large collection of data sets on cell by gene, the CZI platform. That doesn't offer projection right now, but projection of cell types to uploaded data sets, but it may well do, and it comes with a bunch of tools. It has cell ontology, but not down to the sort of depth of annotation that we'd like so far, but we are actually actively working with them to deepen that as well. So I think it's a matter of, there's going to be a whole load of platforms out there that are offering automated annotation in some form, or algorithms that you can use to suggest blast-like algorithms that you can use to suggest annotations. And I think the initial phase is a lot of manual annotation if these things are not originally identified, defined by data. And I think the definition by data is something that's happening mostly in the nervous system. I think elsewhere, if you look at the kidney cells, the new kidney cells found by single cell analysis, you can pretty much pin them down to, here's a new population in a particular part of the loop, or so you can define them in other ways and come up with a set of markers, and you don't need this necessarily a transcriptomic based definition. It's just becoming the norm in the brain. And then for the fly brain, actually, it's more like morphology has become the standard for definition. But I think we're going to build up a set of gold standard annotated data sets, and there's going to be a whole lot of options out there to project this stuff, and I think we need to get ontologies in there into the gold standard annotations so that we get ontology annotations projected and not just free text.
So but also tools would then interact with a provisional cell ontology in one way or the other, is that?
So we have plans to get the provisional cell ontology integrated into Azimuth and into cell by gene. I'm sure we can get it into the single cell atlas at the EBI as well, and they actually do some prediction in the EBI single cell atlas system as well. So I think it's just a matter of getting them out there.
Thank you.
Sure.
So David, I was interested in your idea or your suggestion that complicated cells with complicated labels should be given a symbol. And I think that can work in some cases, but I'm concerned that the symbols themselves might be too abstract for people to readily remember, except in certain cases where they're already in use.
I think it's sort of, where you already have a set of symbols that are in use by a particular community. You can reflect that in synonyms, and there's been talk over the years about having community scoped labels, because obviously different communities will use different ways of referring to things, but there's never really been that much work on community scope labels. And I think we've got sort of good precedent with, if you look at genes, most if you look at all the genes on FlyBase, there's a full name, which is not used too often, and then there's a symbol. So engrailed would be EN, for example, and it's the symbol that's used most of the time. So, I mean, there's definitely challenges because, you know, if you say something like, you know, AD1 or something, you'll find AD1 could be a part of the tracheal system. It could be part of the nervous system. It could be a muscle. You know, these small abbreviations turn up in lots of different places, but then, you know, maybe the context is enough to disambiguate. Yeah, I mean, you know, the shorter those things get, the harder it is to kind of make them unique. But maybe we need to give up on uniqueness and make that context dependent as well. I don't think there's a perfect solution, but I think we need to get closer to what the users expect.
Sure, though, I mean, obviously, I assume that people who are developing, who are analyzing, for instance, single cell seq data, understand that as you get more granular cell types potentially identified and defined, there's probably not a simple way to refer to them. I mean, it's just...
but most people aren't working on every single system. You know, most people who work on the lung don't work on the kidney. So if there's a, you know, if there's some clash there, then it doesn't matter to them. You know, I think.
Sure. I mean, it's, you know, it's a matter of how do we ensure that that we don't get, you know, important clashes that end up being confusing.
You know, but then maybe the tooling needs to cope with those clashes, you know. So if the search allows you to, search gives you a little bit of context. If the search always returns you the full name as well, and the full name makes it obvious that one case is a muscle cell and another case is a neuron, then it's not a problem. Yeah.
Yeah. So I mean, there's an example, for instance, from the area of dendritic cells where at an earlier point, like 15, 20 years ago, the shortcuts DC1 and DC2 were referring to conventional dendritic cells and plasmacytoid dendritic cells. And then later on, a different group of people proposed DC1 and DC2 for two different types of conventional dendritic cells. And so this
yeah, I mean,
it's confusing.
Yeah, I mean, terminological drift is a nightmare, and we have a huge problem in flies where probably the dominant group in fly optic lobe development basically changed the whole nomenclature that we use for the optic lobes. And you know, we took advice from senior people in the field at the time, and they said, no, you should use the traditional nomenclature. And now we have massive, long, extended comments on every term where there's a weird clash, you know. But yeah, I mean, terminological drift happens, and we have to deal with it. Yeah, names with numbering systems that mean this, you know, that refer to different cells are the biggest nightmare. But it's a nightmare for synonyms as well. You know, I don't think it's just an issue with. So probably we should avoid those for the symbols, but then we still need to reflect them in synonyms and maybe have comments, and you know,
sure,
people will search for them.
Are there any more questions or comments for any of the speakers? If not, I think this was actually a very nice set of talks that were in many ways complementary to each other. And I thank all the speakers for participating, and I will post the recording later today on the website. And if the speakers would like to submit their slides to me, I can post those as well. I'll leave that up to the speakers if they like to do that. And so I thank you all, and hopefully we can do this again another year.
Thanks, Alex, for organizing, and
Sure.
and Oliver.
You're welcome.
Yeah, great. Wonderful meeting. Thank you. Thank you, Alex, for organizing this.
Okay, goodbye.
Thanks, everyone. Bye.